Okay, so it's my pleasure to welcome the last but not least keynote speaker of this conference. So it's Jacques Serizay, who is currently a postdoctoral research associate at the Institut Pasteur in Paris in France. So his domain of expertise, of scientific expertise, is spatial organization and regulation of genomes. And of course, as a researcher, he publishes these arguably slightly boring research outputs that are papers. But here in the community, he's known for, I don't know, maybe more exciting or more useful outputs that are very much appreciated here. And among these is the online book Orchestrating High-C Analysis by Conductor. So one of the many, or one of the few high-quality online books that we have and that I think are really helpful for the community at large. And that book is itself the output of another of his really useful contributions, which is the EuroBioC book, which is a package that allows you to write content as a researcher and educator, and then publish it as an online book, but also publish it as containerized or kind of Docker containers or version Docker containers that allow you to reproduce all the analysis in these books with the release or the development version. Right? So very important contributions for the community at large. But that's not quite what he'll be talking about today, as far as I know. So he'll be talking about enhancing genomic research with community-driven and flexible software. So Jacques, thank you very much for being here.
All right. That wasn't me. Okay, yes. So hi everybody, thank you for being here this morning. Would that work? Let's see. Yeah. Okay, so thank you Laurent for the very kind introduction. I am going to give a very brief word about EuroBioC book, but I think it's just 1 or 2 slides. But the rest, yes, is going to be about sort of these somewhat generic title. The shorter version is, this is not going to be a single cell talk, nor a spatial transcriptomics, nor, so yeah. Hopefully something a little bit different and on a topic that I typically work on a daily basis, which is more related to chromatin organization and, as Laurent stated, spatial chromatin organization. Okay, and one of the reasons why I'm not going to talk about single cell is because my data is not really highly heterogeneous. I typically work with yeast, and there's not much to do on a single cell resolution here. Okay, and this is my roadmap, so it's pretty not specific still. So rather than having introduction and then results, package, conclusions, I think I'm going to talk more about the different values that I try to keep in mind whenever I work with bioconductors packages, and starting with this all for one and one for all, which I think to me is the most important one when it comes to the bioconductor community. So in French it would be tous pour un et un pour tous, but the English version is pretty nice as well. And as an example, I'll be talking about the Tidiomics project. So I was curious to see in the room how many people would have the package, Tidiomics, the package installed on their R library folder. Yeah? Some of you? Ooh, okay, yeah, nice. Okay, I have doubts that you have the actual Tidiomics package installed, but we'll see what that means later. Yeah, so the Tidiomics, so I see that a lot of you already are familiar with the idea of Tidiomics. It's an ecosystem, just like Tidyverse would be an ecosystem of functions. And what it does to the bioconductor end users is enabling tidy operations for omics classes. So just like you would manipulate a rectangular data frame in base R by loading additional so-called Tidyverse packages such as dplyr and tidyr, then you can sort of reuse these verbs, but also additional verbs that are dedicated to really these classes that we have available in Bioconductor and that we tend to use in love. So why do we need tidyomics? Well, the answer is in gray, because this is more of an opinion, just like tidyverse. Some people tend to, say this is really, really needed in R. Some say, well, we can do everything in R without tidyverse. This is entirely true. I personally believe that it makes my life easier. So you'll get to choose whether to use it or not. But the more important thing is how did that happen? How did this tidyomic project happen? And that's the important point here. It's been a really community-driven project, mainly spearheaded through open challenges. So if you go to the tidyomics GitHub organization webpage, there is a tab with these challenges listed, and you can scroll and be like, oh yeah, I'm interested in summarized experiments. How do I contribute to this? And it's really super open. There are labels for good first contribution if you're a new developer or new to the world of tidy operations. So I would highly recommend you guys to go and check the tidyomics GitHub repository. Okay, so tidyomics, as I said, it is a package. It is not super highly ranked. And this is because it has been out there only for a year and a half, the package itself, the tidyomics package. But you can install it now. And just like the tidyverse package works, the tidyomics package is an umbrella package. So what it actually does is installing a collection of different packages. And these are the real big players. The tidyomics doesn't provide any function per se. But yeah, you have those different packages that get loaded as soon as you do a library tidyomics, and yeah, maybe here there are the names of the packages that you may be using on a regular basis, or maybe not yet. So ply ranges was, I think, the first sort of effort to bring tidyverse data manipulation to S4 classes, focusing on genomic ranges, and it has been out there for 7.5 years now in BioC. Super helpful. Tidy summarized experiment, out there for 5 years now, and nicely, because we all know that it's a summarized experiment is kind of the cornerstone for Bioconductor. So now we built up on that, and we have tidy single cell experiment, tidy spatial experiment. We also support all the types of omics with and containers with Seurat, tidytof, and recently there has been some effort to allow tidy manipulation of the S4 data frames, which is pretty nice now. Okay, and I'm going to talk about ply interactions, which is my personal contribution, or one of my personal contributions to the project, and a brief word on why ply interaction was a good fit within this ecosystem is to study chromatin conformation capture. So this is an experimental technique that people like me in the lab tend to do to capture pairs of genomic loci in physical proximity. So you can do that in microbes, in single-cell organisms, but you can also do that in individual cells in a tissue or in cell culture, whatever. And that through a series of clever enzymatic reactions that allow you to really capture these chimeric DNA fragments representing physical proximity between two genomic loci that are distant in one dimension. All right. And so this data, which often comes from high-C protocols, but really the generic term would be chromatin conformation capture. This data is often represented at the end by these pairs that can be aligning to different loci along the chromosome, maybe someone even sometimes spanning different chromosomes. And these interactions, they naturally fall within these pairs file representation, right? It's a text-based, rectangular, tabular format where you have an identifier for the pair, and then chromosome 1 start and chromosome 2 start and. So quite natural, and it mimics what I think is. So the pairs file is a format itself, but it's pretty close to what we can know as BedPE format, really. And in R, there is a nice class that has existed for, I think, about 10 years now that allows one to represent these type of pairs. It's called G interactions that naturally store genomic interactions. And it works as follows. You have one anchor, one so-called anchor, and then a second anchor, GR2 here, and then you can wrap them together into a genomic interaction, and that means, okay, well, this bit here interacts with this bit here. And it's very, very helpful when it comes to work with chromatin conformation capture data in R. But the syntax of, so creating these interactions is pretty easy, but then you may want to, change specifically this side of the interaction, or this side of the interaction, or maybe just the start of the second anchor of these interactions. So these operations start then be, they're easy, they're doable with genomic interaction package and the rest of bioconductor pre-existing solutions. But I thought, why not try and adapt the tidy way of doing things for G interactions. And so now what you can do is, so this ply interaction package adapts tidyomics grammar, so a buildup of a tidyverse grammar with added verbs for genomic ranges and so on. And this means you can import, millions of genomic interactions listed in a pairs file with a very efficient package to import a huge file in memory, and then seamlessly coerce that into a G interactions object. So you don't have to first do a, coercing of your anchors, of your first anchors, and then second anchors, and then wrap that. This mimics these as tibble, as data frame, as the G ranges, and now we have as G interactions. And then from there, you can do any of these sort of genomic arithmetics operations. So if you want to work on anchors 2, you can declare that and then explicitly say, oh, I want to get the flanking region downstream, and then I'm now switching to the first anchors, and so on and so forth. And then you also have, so you can tweak these both extremities, but you can also do, very nice subsetting, filtering of your genomic interactions of interest. And I happen to use this ply interactions package on a daily basis now, just because I was already using G interactions objects, and this is sort of an added layer that facilitates my life. So hopefully you guys are inspired by this, and you can go to the tidyomics GitHub repository and look for any open challenge, or if you have your own idea or your own use cases, then we're super open to have you on board. Right? So that was for all for one and one for all. And now I'm going to switch to standing on the shoulders of giant. I haven't said that, Newton said that, or supposedly. And this is another important notion that we have that we cultivate in bioconductor is the reuse of existing classes. Okay. So coming back to my Hi-C workflow, whenever I generate Hi-C or chromatin conformation capture data, I talked about these pairs files, but generally, because they're very huge and we may not need the base pair resolution, what typically happens is researchers, they tend to binarize the chromosomes and compress their data into these essentially sparse matrix formats. So you lose the base pair resolution. Now you have maybe 1 KB resolution, and this can be used to represent interactions as a 2D symmetrical matrix. Okay, and these, for some historical reasons, these formats for Hi-C have been defined as CRUD format and Hi-C file format. So they're binarized data. They're either absolutely no API, or a cron API for Hi-C files to parse the data into R, which historically has really limited the analysis of Hi-C in R. And this is where Hi-C experiment comes up. So it's a solution that defines a series of pointers, so cool file, Hi-C file, and so on, that can be directed to disk-stored files that you would have generated after Hi-C pre-processing. And using, again, reusing the existing import methods provided by EuroBioC IO, one can import some of these data, not the whole entire files in memory, but specifically say, I want to import the data on maybe a specific focus of interest at a given resolution. And you can do the same for pairs files as well, for example, leveraging Ply interactions. And eventually you end up with a Hi-C experiment class, which is here somewhat represented like a summarized experiment object, but it actually isn't really. It's a wrapper, so it reuses the concepts of summarized experiments, but because it doesn't store square data, what it does store is interactions. I couldn't actually reuse directly the class itself. Unfortunately, I tried and then it was odd. What I did try to reuse as much as possible is, so these genomic interactions, they're the core of this object, together with simple lists for different scores, and then again, simple lists provided by S4, vectors for topological features and so on. And so this, ideally, as much as possible, I tried to at least, make this object feels as natural as possible for the bioconductor end users. So anyone who would have used summarized experiment classes or genomic ranges or genomic interactions, they should feel natural with this type of class. And I think that's, talking back about the, standing on the shoulders of giants, I think that's the reason why we tend to be able to develop and implement new features fast, is because the end users are happy with these new features. They don't feel so new, they feel like natural. Okay. So this Hi-C experiment package is now acting as a sort of a intermediate between disk-stored files, and I'm also leveraging the EuroBioC file cache infrastructure to get access to a number of public repositories for Hi-C and cool files. And it interacts then with Ply interactions in high contacts to perform sort of, entry-level or intermediate or somewhat advanced Hi-C and chromatin conformation capture analysis. Okay, and so that has been really helpful to accelerate my research. But one thing is that I would outline is the research that I'm lucky enough to do is not restricted to a single organism, right? I work with yeast, I work with bacteria and phages, with eukaryote parasites, with mouse. And this is, I think, something that is not really entirely fully appreciated in bioconductor ecosystem, is we're not restricted to human or mouse or whatever. The methods that we implement, they're generic enough to work on any organism. Again, that's one of the important points to me in a bioconductor. Okay, and because I felt that there was room for it, as Laurent has stated, I wrote this orchestrating Hi-C analysis with bioconductor book that contains a lot of introduction material and then sort of in-depth analysis and eventually some more advanced topics with workflows. So some content, and then to facilitate this writing, what I wanted to do is reuse what had been initiated with the orchestrating single cell analysis book, which makes a good use of this vignette folder with a make file in there. And what I did was formalize this implementation within the EuroBioC book package. And so if you install this EuroBioC book package, in 4 lines of code, you can get a working package, a working book documenting whatever you want to document, published within your own GitHub account, available on your own GitHub pages, that can also has a matching Docker image with all the dependencies automatically listed from your description file. So this installs a package that then gets sort of built into a book. And because this is compatible with the bioconductor build system, then you can, you work on your book, you write your quarto markdown pages, and eventually you say, well, I actually would like to have that available within bioconductor. I'm happy with the development I've made and the writing I've made so far, but now it's time to put it out there for the bioconductor community, so you submit it to bioconductor through the contributions repository, and then the BBS tries to build this, and it says, well, nice. No problem, I can build it, there's run series of checks, BioC checks, no errors. So yeah, it's valid for, green tag, okay, no errors, and it can be accepted and put within the bioconductor own repository. And what happens when the BBS actually build that book is it renders, through this MEC file, it renders the quarto markdown into a book and stores it into doc here. So eventually, when you submit it to the bioconductor ecosystem, eventually what happens is you have it on your personal GitHub account, but you also have it, then every new version of BioC will automatically bump your package version and your book version, and will regenerate a Docker image associated for every bioconductor version that you have. So if you have an idea of some cross-package documentation that you would like to write, I would highly encourage you to use BioC book, and particularly this enables you to get a Docker image for reproducible content. All right, and the last bit is about this sort of definition from Thomas Huxley about science being nothing but trained and organized common sense. Ideally, when you read a methods paper, or even a methods section of a paper, you see that the authors say, well, we've done the experiments, and then we've pre-processed the data this way, and it was no problem, and then we did the analysis this way, or at least the bioinformatician told us that they did that this way, and so it looks easy, right? It's like, whew, just a single line. For me, it looks a little bit more like this, where you have your original files, and then actually you realize that maybe the genomic sequence you were using wasn't so good, so you have to switch to a new sequence, which means also you have to reprocess all the data that you have aligned to your original genomic reference. Maybe you have duplicates, maybe they're actually resequencing. It starts getting messy at some point. And the more you work with collaborators, the more you have this mess. But perhaps it's just me. Perhaps I'm just not great at organizing my data. But still, what I would love to have is some intermediate space here to consolidate my data in a sort of programmatic way to ensure that everything is at the very least, aligned to the genomic reference that I'm using when I work maybe on a certain strain of yeast or a certain mouse mutant or whatever. And that's where Momix comes in, which is a software that I've started writing for a few months now, which is a tool to manage collections of genome-wide data sets. And mostly Momix provides you with some ingestion and extraction command line, subcommands that would allow you to either put these files within a single repository, within a single Momix repository, or extract them back from this Momix repository. Okay, and you can store these different different modalities of data, and this Momix has then support for cloud access, and eventually, I think, it's later. Yeah, eventually you can use this Momix for backup, for sharing, and for cloud access. So just going back, how does this work internally? The Momix repository relies on a number of TileDB arrays. So it's sort of like a ZAR cousin, but with pros and cons compared to ZAR. And it's supporting multimodal and high-dimensional arrays natively. So you can ingest bigwig files or FASTA files or genomic features, and you can put them into these high-dimensional arrays. And that can be done through either manually, yes, or momics. So how does that compare to traditional storage with big wig files? Well, the more you ingest, the closer it gets to the storage space taken by big wig files, but you still have almost one order of magnitude of storage footprint gained through momics compression. And for FASTA sequence, it's not as efficient. It's in between a zipped and unzipped FASTA file if you store a genome. So maybe a momics repository with a mouse genome sequence would be maybe 1 gigabyte. Oh, it's actually here. Yeah, about 1 to 2 gigabytes. But it already stores it in a one hot encoded version. That means that it's ready for bioinformatic and machine learning applications. And so what this means is, after that, when you have your momics repository, you can do queries and you can stream the data using this query API, and then the features, they don't become, is it a sequence, or is it a genomic track, or is it genomic features? No, it's just any type of feature. So now you have a unified API to query multiple modalities, multiple data modalities. And I'm going to go a little bit faster now to finish on time, but just to outline that this tool performs 1 to 2 orders of magnitude faster than PyBigWig. It's also quite faster than PyFA index to query genomic sequences. And then you can use those momics repositories to form TensorFlow compatible data sets in order to get input features and output labels from a momics repository, and eventually train these models. And so in order to perform multimodal feature extraction from different types of modalities that have been ingested in your momics, perform learning joint representation, and eventually perform multitask learning. Okay, so I'll skip over the use case just to show that you can use momics to do actual biological research. And I think I'll finish with this call. So momics is actually written in Python, but because the talk stated community-driven, here's a call to anyone who would be interested in working either with me or independently. I'm happy with that, to work on a BioC API for momics, particularly to implement support for genomic ranges and RLE, etc. With that, I'll briefly mention the different people I worked with: Romain, Jessica, Julien, and Alice, and then lots of others not listed here. But and I'll leave you with this note on different values I hold dear. Thank you.
Thank you very much, Jacques. So we have time for questions.
Sorry. Just wondering, behind the hood, when you're actually streaming, are you using SAM tools? Are you calling other tools, or is this from bottom up?
Yeah, it's entirely different architecture. So the files, the way the files are consolidated is you don't have BAM files, you don't have bigwig files anymore. Sorry, you meant about momics, or? Yeah, yeah. So these files are ingested in a different, in a compressed array format. So you use, under the hood, it's the TileDB R library, well, or Python library in this case, that is used to query TileDB arrays. So there's no dependency in any way for SAM tools or PyFA index or PyBigwig or whatever.
Any other questions? Yes.
Yeah, so I was going to ask something about the TileDB as well. I don't quite remember how they were realized in the disk, but do you guys have separate, folders for each individual TileDB array? But are you using the native hierarchical TileDB format? How do you do that?
Yes, so here it's represented as folders, the genome, coverage, and annotations. But there, when you ingest the data, the way I implemented it is these are hierarchical folders supported by TileDB. So they're essentially nested TileDB arrays. Yes. And then the nice thing is you can either query the whole subarray, the whole subdirectory, or individual arrays with chromosome 1 only or chromosome 2 only.
I have a question, or maybe kind of starting something. So the tidyverse, tidyomics, the goal is to have this umbrella package that then opens up various possibilities. But the idea is to have kind of, a package to combine things. In Bioconductor, we have lots of documentation. Arguably somehow too much, overwhelming. We have vignettes, we have how-tos, we have books, the man pages. Do you think it would be possible to apply the same concept of a kind of umbrella feature to try to pull together different types of documentation for maybe a specific topic?
I'm not sure I fully understand. So you would use sort of tidy operations to aggregate documentation?
No. More the concept of some sort of umbrella documentation
I see.
to try to pull a certain set of documents on the topic to really help users. They don't need to know all these different places or different packages to look for the information.
Yeah, so I think at least in my experience writing this bit of resources, it indeed comes very useful when you have a number of packages, and indeed what you want to do is highlight the interoperability between packages, not because for individual package, indeed you have vignettes and man pages, and this is enough already, I think. But yeah, when it comes bigger, you would need cross-package documentation. So I think BioCbook is a good place to start. But then to do that in a programmatic way, that's an interesting point. I don't think I've ever thought about it before. Maybe there's a way to, I don't know, collapse all the different vignettes into a book. But I think the manual authoring of ideally a collection of authors for a book is better than just aggregating different bits of resources together. But I agree, there's definitely some room, some work to do on aggregating knowledge.
Okay, thank you, Jacques. The MOMICs, thanks for telling me about that. That's pretty interesting. One of the things that I think Bioconductor really grows from are exemplars, like TCGA for the multi-assay experiment and the idea of taking a really rich multi-omic and multi-phenotypic resource. Do you have any ideas of exemplars that would get people really excited about MOMICs for integrating and interrogating a high-value genomic resource?
Yeah, so for absolutely, I think that's a very interesting question, and I thought about it to sort of sell the product. So yeah, implementing, so the one use case I had in mind is to ingest the encode and mode encode number of data sets. I think that it builds up to several thousands of genomic tracks, even more for genomic features. And so this is a nice place to start to show that it can scale up to thousands of data sets and illustrate that you can do that for different organisms.
Yeah, that's a good one. So like a curated encode, right?
Yes.
Yeah.
And mode encode. And model organisms are important.
Yeah.
Yeah.
So that would be a really nice exercise.
Yeah.
Be great to think of some other examples, but thanks.
Well, and personal usage is I've already parsed most of the ChIP-seq experiments that have been done in yeast, because I typically work with these. So, and these MOMICs repositories then can be stored on a AWS server and then be queried directly.
And maybe there was a last question here.
Thank you, Jacques. Really nice talk. I also have a question regarding the genomics package that you mentioned about. You talked about the revolutionary way of storing multi-omic data. I was wondering, is it a kind of compressing like files in a different, using different algorithms, or also I was also wondering to know which kind of level of data is it raw data that you can compress, or like
so regarding the first question, the way the data is compressed per se, specifically talking about compression, they use standard compression algorithms. Now how they wrangle the data is, how TileDB wrangle the data is a little bit different. They put it in these TileDB arrays, which are multi-dimensional arrays. So it's one way to store data on a disk. That allows you a lot better compression than a text file, for example. So that's for the first question, and the second question was about, sorry, I forgot the, come to me.
I was wondering which kind of level of data that you store, if it's raw data or processed data.
So it could be virtually anything. So far what I support, what I implemented is it sort of here? No. It shows up here. I have a genome for a genomic sequence. I have coverage, so they're essentially big-wig tracks, processed files, annotations. They could be somewhat raw data, but not really. But you're welcome to add anything. So an additional type of modality that I'm going to implement is variance, because we have a pretty good usage of variance in genomics. But then for raw data per se, like FASTQ files, for example, that's not really the purpose of this. Because it's meant for sharing, particularly in my mind at least, processed data, for reusability of processed data, then that's how I see it being used.
So in the interest of time, I thought I saw another hand, but please go to Jacques to ask more questions during the next break. Thanks again, Jacques, for your presentation.